Hello students, today's lecture is on the types of immune response for clonal selection theory. Now let us have a brief introduction. A mature immunocompetent animal contains a large number of antigen-reactive clones of T and B lymphocytes. The antigenic specificity of each of these clones is determined by the specificity of the antigen binding receptor on the membrane of the clone's lymphocytes. The specificity of each T and B lymphocyte is determined before its contact with antigen by random gene rearrangements during maturation in the thymus or bone marrow. The role of antigen becomes critical when it interacts with and activates mature antigenically committed T and B lymphocytes, bringing about expansion of the population of cells with a given antigenic specificity. In this process of clonal selection, an antigen binds to a particular T or B cell and stimulates it to divide repeatedly into a clone of cells with the same antigenic specificity as the original parent cell. Clonal selection of lymphocytes is the central principle of adaptive immunity. Remarkably, at the time that Burnett formulated his theory, nothing was known of the antigen receptors of lymphocytes. Indeed, the function of lymphocytes themselves was still obscure. Lymphocytes did not take center stage until the early 1960s when James Gowans discovered that removal of the small lymphocytes from rats resulted in the loss of all non-adaptive immune responses. These immune responses were restored when the small lymphocytes were replaced. This led to the realization that lymphocytes must be the units of clonal selection, and their biology became the focus of the new field of cellular immunology. Clonal selection of lymphocytes with diverse receptors elegantly explained adaptive immunity. Burnett proposed that developing lymphocytes that are potentially self-reactive are removed before they can mature, a process known as clonal deletion. Clonal selection of lymphocytes is the single most important principle in adaptive immunity. The specificity of receptors is determined by a unique genetic mechanism that operates during lymphocyte development in the bone marrow and thymus to generate millions of different variants of the genes encoding the receptor molecules. Thus, although an individual lymphocyte carries receptors of only one specificity, the specificity of each lymphocyte is different. This ensures that millions of lymphocytes in the body collectively carry millions of different antigen receptor specificities, the lymphocyte receptor repertoire of the individual. During a person's lifetime, these lymphocytes undergo a process akin to natural selection. Only those lymphocytes that encounter an antigen to which their receptor binds will be activated to proliferate and differentiate into effector cells. This selective mechanism was first proposed in the 1950s by Macfarlane Burnett to explain why antibodies which can be induced in response to virtually any antigen are produced in each individual only to those antigens to which he or she is exposed. He postulated the pre-existence in the body of many different potential antibody-producing cells, each having the ability to make antibody of a different specificity and displaying on its surface a membrane-bound version of the antibody that served as a receptor for antigen. On binding antigen, the cell is activated to divide and produce many identical progeny known as the clone. These cells can now secrete clonotypic antibodies which is specifically identical to that of the surface receptor that first triggered activation and clonal expansion. Burnett called this the clonal selection theory. Now let us see the postulates of the clonal selection theory. Each lymphocyte bears a single type of receptor with a unique specificity. Interaction between a foreign molecule and a lymphocyte receptor capable of binding that molecule with high affinity leads to lymphocyte activation. The differentiated effector cell derived from an activated lymphocyte will bear receptors of identical specificity to those of the parent cell from which that lymphocyte was derived. Lymphocytes bearing receptors specific for ubiquitous self molecules are deleted at an early stage in lymphoid cell development and are therefore absent from the repertoire of mature lymphocytes. Now let us see the characteristics of clonal selection theory. During clonal selection, the number of lymphocytes specific for a given antigen is greatly amplified. Moreover, many of these lymphocytes, referred to as memory cells, appear to have a longer lifespan than the naive lymphocytes from which they arise. The initial encounter of a naive immunocompetent lymphocyte with an antigen induces a primary response. A later contact of the host with antigen will induce a more rapid and heightened secondary response. The amplified population of memory cells accounts for the rapidity and intensity that distinguishes a secondary response from the primary response. In the humoral branch of the immune system, antigen induces the clonal proliferation of B lymphocytes into antibody-secreting plasma cells and memory B cells. The primary response has a lag of approximately 5 to 7 days before antibody levels start to rise. This lag is the time required for activation of naive B cells by antigen and Th cells for a subsequent proliferation and differentiation of the activated B cells into the plasma cells. Antibody levels peak in the primary response at about day 14 and then begin to drop off as the plasma cells begin to die. In the secondary response, the lag is much shorter, only 1 to 2 days. Antibody levels are much higher and they are sustained for much longer. The secondary response reflects the activity of the clonally expanded population of memory B cells. These memory cells respond to the antigen more rapidly than naive B cells. In addition, because there are many more memory cells than there were naive B cells for the primary response, more plasma cells are generated in the secondary response and antibody levels are consequently 100 to 1000-fold higher. In the cell-mediated branch of the immune system, the recognition of an antigen-MHC complex by a specific mature T lymphocyte induces clonal proliferation into various T cells with effector functions, Th cells and CTLs, and into memory T cells. Now let us see how do we know that clonal selection hypothesis is largely correct. The binding of an antigen to an individual lymphocyte can be visualized by labeling. Using this approach, experimenters showed that one lymphocyte could be bound by one antigen, but never by many. Also, antigens can be radioactively labeled, and when such antigens bind their specific lymphocytes, the latter are killed by the radiation. Still, the animal that received the injection of the radioactive antigen will be perfectly able to respond to a wide array of other antigens, indicating that only the clone specific to the radioactively labeled antigen was killed. Finally, proteins that make up the antigen receptors have been examined in detail. The amino acid sequence of these proteins, as well as the sequence of nucleotides in the genes that code for these proteins, has been solved. These studies show that individual antigen receptors have unique combining sites, the parts of their molecules that combine directly with the antigen. Any two different lymphocyte clones will always have two distinct antigen receptors, each with its own amino acid sequence at the combining site. Taken together, this entire evidence supports Burnett's hypothesis very strongly. If antigen encounter stimulates proliferation of lymphocyte clones, will an immunization or infection lead to massive increase in the number of blood cells? The answer is no. We have to realize that at any given moment of time, a vertebrate organism has at its disposal at least 109 different lymphocyte clones, each with its own specificity. The invasion of any individual antigen will cause proliferation of the clone that is specific to it exclusively. Some antigens, for example, large protein and polysaccharide antigens, have more than one epitope, that is, more than one site that can be bound by antibodies or TCR. In this case, several different clones may react to the antigen, each of them to a separate epitope. The non-specific clones will not be affected, however, therefore. No matter how strong the proliferative reaction is, the overall increase in the number of cells in the blood will be negligible. Suppose two clones react to two distinct epitopes of an antigen and each clone expands 10,000-fold. The overall increase in the number of cells will be 20,000, which is still a negligible increase. That is 0.002% over the number of all lymphocyte clones, 1 billion. Now let us see the main practical significance of the clonal selection hypothesis. Perhaps it is the development of the hybridoma technology. Based on the principal postulate of the clonal selection hypothesis that lymphocytes exist in clones, each clone possessing its own antigen specificity, G. Kohler and C. Milstein in 1975 discovered a way to immortalize clones with particular useful antigen specificity through their fusion with myeloma cells. The resulting hybridomas can be used and are very extensively used for production of useful monoclonal antibodies as well as for studies on antibody genes. The impact of hybridoma technology on basic science, medicine, pharmaceutical industry, agriculture, etc. turned out to be so big that in 1984, Kohler, Milstein and Jerin were awarded Nobel Prize. Now coming to the conclusion. Because we can make hundreds of thousands, maybe even millions of different antibody molecules, it is not feasible for us to have too many lymphocytes producing each type of antibody. There would not be just enough room in the body to accommodate them. To compensate for this, lymphocytes which are triggered by contact with antigen undergo successive waves of proliferation to build up a large clone of plasma cells which will be making antibody of the kind for which the parent lymphocyte was programmed. By this system of clonal selection, large enough concentrations of antibody can be produced to combat infection effectively. The importance of proliferation for the development of significant antibody response is highlighted by the ability of antimitotic drugs to abolish antibody production to a given antigen stimulus completely. Because it takes time for the proliferating clone to build up its number sufficiently, it is usually several days before antibodies are detectable in the serum following primary contact with antigen. The newly formed antibodies are a consequence of antigen exposure, and it is for this reason that we speak of the acquired immune response.